Cell-mediated immunity. Cellular immunity is a specific acquired immunity. It is accomplished by the effector T cells and the macrophages. All the cellular elements which are involved in acquired immune response, they originate from these pluripotent stem cells which are present in the bone marrow. Now they can have two modes. One, they may migrate from the bone marrow into the thymus and then develop into the T cell, or they may continue to remain in the bone marrow and then form the B lymphocytes. So this bone marrow and the thymus are together known by the name of primary lymphoid organs. The cell-mediated immunity is generally invoked for dealing with the organism which prefer to stay within the cells of the host. So they may be the viruses, the fungi, the intracellular bacteria, for example, Mycobacterium leprae, tuberculosis, or even brucella. So the cell-mediated immunity may also be involved in graft rejection or the allograft rejection, or they may be also responsible for a delayed hypersensitivity reaction which is seen after several days. The immature T lymphocytes enter the thymus from the cortex and then they will leave through the medulla. So as they travel from the cortex, they come across these large epithelial cells which are known by the name of thymic nerve cells. So then the T cells, they undergo the process of differentiation and there is reorganization of the DNA which results in the characteristic formation of T cell receptors or the surface markers. So then in addition to this, the cell will also develop MHC restriction, and if it is able to recognize the self MHC, it will go to the next step. If it is not, then it will undergo the process of apoptosis. So next, the TCR recognizes the self antigen. If they are recognizing the self antigen and reacting with that, then they will undergo the process of apoptosis. Else they will be develop self tolerance and then they will move into the corticomedullary junction. In the corticomedullary junction, the lymphocyte will meet the macrophages. There will be interaction between the macrophages and the lymphocyte, which will mount an immune response to the cell. So the T lymphocytes will then develop a capacity to not self-react but recognize the antigen which are present in the body, and then they will move out from the medulla into the blood vessel and then it will be carried to the different secondary lymphoid tissues which are present in the body. The differentiation of the precursors of T cell is associated with the reorganization of DNA, which results in expression of characteristic T cell surface markers, as you can see here, the rearrangement process. So another ability that is developed in the cell is the development of MHC restriction. That means their capacity to respond to get limited to just one genetic variant of MHC molecule. So these aspects of differentiation are accomplished by the various epithelial cells of the thymus. The tolerance to the self also develop in the thymus, and it seemed to result from interaction between the macrophages and the lymphocytes, which are likely to mount an immune response to the self. So these are the lymphocytes which have a greatest affinity for macrophages. So what happens if a T cell is mixed with the body cells with different self antigen? As you can see in this figure, the T cell is being mixed with the different type of body cells with self antigens. In case there is a reaction between the T cell and any of these, then the T cell will be eliminated, or it will be taken up by phagocytosed and it will be destroyed. So no self-reacting T cell will be released into the circulation. So this phenomena is known by the name of self-tolerance. Now the thymus is just like a school where the immature T cells are provided with the characteristic protein which provide them an identity, and also they provide capacity to perform certain functions. The thymus also conduct an exam for these T lymphocytes to ensure that they are fully qualified and safe before they leave the thymus. So in absence of thymus, the response to the cell-mediated immunity is not eliminated, but it is rather diminished. There is a syndrome by name of DiGeorge syndrome in which the structures which are derived from the third and the fourth pharyngeal pouches do not develop, including the thymus and the parathyroid gland. So these persons, because of the diminished cellular immunity, will suffer from the opportunistic infections such as infection from the fungi and even the viruses. The T and the B cell populate the secondary lymphoid organ, which may include the lymph node, the spleen, the lymphoidal tissue associated with gut, respiratory tract, and the mucosal surface. So the role of these secondary lymphoid organs is that they trap the organism and mount an immune response. So they may be trapped in the draining lymph nodes or in the spleen or even the mucosal surfaces and mucosa associated lymphoid tissue. So irrespective of the site, the response to the entry of agent will follow a common pattern, that is, cellular immunity will be hosted against that pathogen. The T cell can be classified into four main types. First is the cytotoxic T cell that destroys the virus infected cells and the tumor cell. Another is helper T cell, which assist other lymphocytes to mature and activate. Then we have a suppressor T cells, which modulate the immune system and prevents the autoimmune diseases. And fourth, we have the memory T cell, which help to augment the immune response after the reintroduction of the pathogen. Cytotoxic T cell, they are the killer cells. As we can see in this figure, they destroy the antigen directly by attaching to them and then releasing the chemical perforin to kill them. So these destroy not only the microorganism, but sometimes they are also responsible for destroying the body's own cell. The next type of cell is the helper cell, abbreviated as TH. These helper cells, they attract and stimulate macrophages. They cause increase in activity of B cell to increase the antibody production. Also cooperates with T cell and causes the activation of cytotoxic T cell. So these are the most numerous type of cell, that is 3/4 of the T cells is formed by these helper T cells, and they also release lymphokines which act on the immune system and the bone marrow cell to cause the enhancement of the immune response. The helper T cell is of two types depending upon the type of pathogen which is recognized. It can be helper type 1 or the helper type 2. In helper type 1 cell, it helps in the activation of macrophage and the cytotoxic cell by release of interleukin 2, gamma interferon and tumor necrosis factor B. So this type of cooperation is known by the name of T cell cooperation. Then we have helper type 2 cell, which is responsible for helping in activation of beta cell, which is responsible for production of antibodies. And the interleukin responsible are interleukin 4, 5 and 6 secreted by helper 2 type 2 cells, and then it is known by the name of TB cooperation. The T helper cell causes the release of these lymphokines, and these lymphokines are responsible for enhancement of the immune response by the different cells in the cellular immunity. And without these lymphokines, the T helper cells and even the immune system will almost be paralyzed. Then another function of the T helper cell is stimulation of growth and proliferation of the cytotoxic T cells. And you can see interleukin 2, which is released by T helper cell, causes a strong stimulatory effect on these cells. The next function can be known as autostimulation of T helper cell itself, and that is mediated by again interleukin 2, which can causes a direct positive feedback effect on activating the T helper cells themselves, and therefore it acts as an amplifier. The next function is stimulation of B cell growth and differentiation to form plasma cells and antibodies. Interleukin 4, 5 and 6 are responsible for this action. Then we have the memory cell. The memory cell do not have any action as such, but on getting a repathogen or reinfection, they multiply very fast if it is a second invasion, and producing even a bigger clone of T lymphocyte, resulting in rapid destruction of the antigen, which we can see in this graph also. The first exposure, you can see the peak and then back to the normal. And on the second exposure, you can see the time taken is less and the response is more amplified. Then we have the suppressor cells or the regulator cells. They are responsible for slowdown of the vigorous response of cytotoxic and the helper cells during the mediation of phagocytosis and the lysis of the cells. So the slowing down and stopping of the immune response is brought by the T cell, that is suppressor cells. It acts on T helper, B cell, macrophages, and even the cytotoxic cell. So the various lymphokines which are released, one is interleukin 2, which is causing two action. One, a positive feedback for the helper T cell and also leading to the stimulation of cytotoxic T cells. And the other one is interleukin 4, 5 and 6, that is responsible for the stimulation of B cell. The macrophage accumulation or the activation and enhancement of killing are brought by non-specific lymphokines. Now, the two mechanisms by which cell-mediated immunity operates is by helping phagocytosis and release of certain cytotoxic substances. So, accordingly, the T cell employs two of the cells for this purpose. One are the T helper cells and another are the cytotoxic T cells. Now, when we talk of the stages of the cellular immune response, it occurs in 5 stages: antigen processing, antigen presentation, recognition of antigen by the lymphocyte, T lymphocyte differentiation, and attack phase of the cellular mediated immunity. Now, the activation of T cell is brought about by specific antigen, resulting in the activation of resting T cells into an mitosis phase. However, this activation or the recognition of antigen is brought about by the antigen fragments which are presented by MHC molecules, and they can be the macrophages, the B lymphocyte or the dendritic cells, which presents the fragment of antigen on their surface in association with these MHC proteins. Now, the MHC molecules which are encoded by the major histocompatibility complex are of two types, MHC1 and MHC2. MHC1 is responsible for presenting the antigen to the cytotoxic T cells, here we can see, and MHC2 presents the antigen to the helper T cells. The antigen in the context of MHC is recognized by as many as 1 lakh T cells receptors per cell. The antigen processing is where an antigen is phagocytosed and degraded into polypeptide by the antigen processing cells, which may be B lymphocyte, the T lymphocyte or the dendritic cells. So here in this diagram you can see the antigen has been degraded into fragments of antigen and then they are represented on the surface of this cell and then presented to the lymphocyte. Second step is antigen presentation, in which the antigen polypeptide fragments associated with MHC proteins are expressed on the surface of antigen presenting cells. Here you can see the MHC protein represented in blue color is interacting with the T cell receptor of the T cell, and then this interaction is known by the name of antigen presentation. The second step is recognition of antigen by the lymphocyte. The CD4 cells will recognize the class 2 MHC and CD8 cell will represent the class 1 MHC. The T cell receptors recognize and interact with the single antigenic determinants on the antigen. Now the T cells that has been activated by the antigen presentation process, it now undergoes a proliferation and differentiation to form the cytotoxic T cells and the suppressor T cells. The cells are released into the lymph and circulated throughout the body and then recirculated from the tissue fluid back to the blood. So this process may last for years also, and the memory cells which are formed during this process are helpful in the subsequent exposure to the antigen. Now these cytotoxic T cells bind with the antigen bearing cells or the target cell and destroy them by following mechanism. The first is perforin mediated killing, where the cytotoxic T cell, after binding with the target cell, secrete a hole forming protein which is known by the name of perforin. Here you can see, and then this causes the punching out of the holes in the membranes, and in the presence of extracellular calcium, the pore so forms causes cell death by disrupting the cell homeostasis and then osmosis, and then leading to the fragile cell which is then ruptured. The second can be the lysis through the cytotoxic substances after binding to the target cell, and third, it can be an induction of the apoptosis, where the cytotoxic T cells secrete the tumor necrosis factor which causes the process of apoptosis. The perforin mediated killing by the cytotoxic cell starts with the cytotoxic cell secreting the hole forming protein known by the name of perforin. These punched out holes in the cell membrane of the target cell causes the movement of fluid into the cell from the interstitial spaces, and also certain release of cytotoxic substance and granzymes which are present in the cytotoxic T cell causes the cell to swell and causes the lysis in the infected cell, and this leads to the cell lysis or the cell death. This slide represents the summary of events that occur in cell-mediated immunity. The first step is antigen presentation, that is followed by the antigen processing, and it is done by mainly the B lymphocyte, the macrophages and the dendritic cells. Then there is release of cytokines or the lymphokines, and mainly it is the interleukin 2 which is responsible for the activation of the cytotoxic T cells, which then binds to the infected cell and bring about the cell death by three mechanisms. It can be perforin mediated death or killing, then it can be lysis of the cell, or it can be induced apoptosis.